Hey everybody, let's get started with our third and final blood vessel video. So we're beginning today with capillary blood pressure. We've got a nice continuous capillary right here. Its blood pressure is generally low from about 40 millimeters of mercury at the beginning of the capillary to about 20 millimeters of mercury at the end. And this low blood pressure is great for exchange, which we know is the big job of capillaries. Now, if we go onward to veins, the blood pressure is even lower in veins because we're even farther from the pumping action of the heart. We know the low blood pressure in veins is why they have thin walls, is why they can be located, somewhat superficially. The problem though, is that if we have low blood pressure in our veins, so we know veins need to send blood back to the heart, right? And if there's not very much pressure in the veins, then the pressure difference between these two things is very small. There's a very small gradient driving blood flow back to the heart. However, we do have some tricks up our sleeve that help us get blood back to the heart. And by the way, that movement of blood back to the heart is known as venous return. So getting blood back to the heart. So, like we just said, there is still some venous pressure, and that venous pressure is still pushing blood, and that venous pressure is leftover pressure from the contraction of the heart. So that's going to help us move blood back to our helps, can help venous return. Gravity is a little bit trickier. Basically, if you think about gravity can be either a help or a hindrance in structures above the heart. So in structures above the heart, gravity is going to help us by helping move blood down to the heart. In structures below the heart, gravity is a hindrance because it's moving blood away. So gravity is a little bit of both sides of the coin there. Now there are two other things that are acting to pump blood. There is something called the skeletal muscle pump, and then something called the respiratory pump. We're going to go over these guys. And then there is venoconstriction, which as its name implies is the constricting of the vein. So let's go over all of these things, starting with that remaining pressure. So veins do have some blood pressure to them. There is still something of a gradient between our veins and our right atrium, which on our graph would be over here. So there is still some of that systolic pressure generated by the contraction of the ventricle that is going to push blood forward. But it ain't enough. It isn't enough. So gravity, we know gravity can help or gravity can hinder. Of course, who is going to have better venous return? The gentleman here or the madam? Man or woman here? Guy or girl? Who's going to have the better venous return? Considering that gravity can help us or gravity can hinder that venous return. All right, let's keep going. Let's talk about one of our pumps. There is a pump known as the skeletal muscle pump. And basically what's going on with that is working muscles are squeezing neighboring veins. So here I got two working muscles. As these muscles contract, as their muscle bellies are squeezing, this vein here is getting squished. Blood tries to flow backwards, but hey, valve prevents that. So blood is forced forward. So movement is going to increase venous return by activating the skeletal muscle pump. There's another pump we got. When we take a deep breath in, so here is before the deep breath, here is after the deep breath. Notice that the greenish area, the thoracic cavity, its volume increases. An increase in the volume of a cavity, of a container, is going to cause the pressure of that cavity to decrease. This is good old Boyle's law, which I know you know from chemistry class, whenever that was, however long ago that was. So notice that the heart is in the thoracic cavity. So if we lower thoracic pressure, we're lowering heart pressure. Remember, we're trying to go from veins to the heart. And there is a small gradient there, small pressure gradient there. And if we lower heart pressure, if heart pressure is now even lower, our gradient becomes steeper and we get better blood flow. So by taking a deep breath in, we expand our thorax. Look at that diaphragm. Notice how it goes down. And the thoracic volume increases, making the thoracic pressure decrease, making pressure in the heart and the neighboring veins decrease. And that is going to draw blood back into the heart. All right, finally, for venous return, there's venoconstriction. First of all, can you tell which is the artery and which is the vein here? Quickly, you should tell me this is the artery because you see its circular shape and its relatively thick lumen, not lumen, wall. Over here, my vein has more collapsed and it has a skinnier wall, However, that skinny wall does have muscle in it. There is muscle in here. And when that muscle contracts, that is going to squeeze this vein, raising pressure in here and pushing blood back to the heart. All right, so that is venous return. Notice that if venous return increases, so an increase in venous return means we're going to get more blood going into our heart. And we know the Frank-Starling law says more blood in means more blood out. So an increase in venous return yields an increase in cardiac output. Excellent. With that, we are going to move onward and talk about resistance. Resistance in blood vessels is opposition to blood flow. It is basically a measure of friction between the blood and the vascular walls. When we talk about all of the resistance in our body, we use the term total peripheral resistance. And one of the important things about resistance is it allows us to direct blood flow. All right, we change resistance to direct blood flow. For example, in my friend here, I want the resistance of blood vessels in his leg muscles to be low because that's going to promote flow. Meanwhile, in his large intestine, I want the resistance there to be high to prevent blood from flowing to the large intestine, because if we got more blood going to one place, we got to get less blood going somewhere else. So I'm going to use changes in resistance for the directionality of blood flow. That is an important take-home point. Do not forget that. Don't forget anything. Just remember every single thing I'm saying, and you'll do great on your test. All right. There are three major sources of resistance. There is the viscosity of blood, the total vessel length of all of our blood vessels, as well as the vessel radius. Now, obviously, I bolded, I underlined, I clouded my vessel radius words here because it is the most important for three reasons. Number one, it is changeable. It is hard to change length. It is hard to change viscosity, but radius is changeable. and it's quickly changeable. Let's add that word, quickly. It is quickly changeable. So I can change resistance by changing radius fast. I can't change viscosity or length fast. Number 2, radius is locally changeable. Of course, if I want more muscle to go to my dermis, I need to change the resistance only in my dermis. If I want more blood to go to my dermis, I change the resistance to my dermis, not to other places. So vessel length, there can be local changes for that, but that takes way too long. Viscosity, I can't do any local changes. Lastly, and we'll touch on this in a second, there's a mathematical thing going on where a small change, my triangle there means change, a small change in radius, is going to create a big change in resistance. A big change, there it is, in resistance. I'm just going to write re for resistance there. So 3 reasons why radius is the most important source of resistance, right there. But let's talk about everything. So resistance is directly proportional to viscosity. If viscosity goes up, there's more resistance, there's less flow. Classic example of a viscous fluid is whatever a Wendy's Frosty is made out of. It is super thick, it is super viscous. You're not drinking this with a straw. Okay, it's too viscous. There's too much resistance to flow. Now, we don't have Frosties. Well, we might have Frosties in our body, but definitely not in our blood vessels. In our blood vessels, we have blood, and the major contributor to blood viscosity is red blood cell count. So basically, resistance depends on hematocrit, which is the percentage of blood. You remember this word, right? The percentage of blood made up of red blood cells. Now, usually, viscosity does not change. However, if our EPO goes up, if we make more EPO, erythropoietin, we will increase red blood cell count, which will increase viscosity, which will increase resistance, which will decrease flow. All right, but this is not changeable quickly. This is not changeable locally. All right, let's move on to length. Total blood vessel length. So longer blood vessels have more area. More area means more friction. More friction means more resistance. So increased length means increased resistance. We can grow new blood vessels. There can be angiogenesis, especially if we grow fatter. If we grow fatter, we grow new blood vessels in that adipose tissue. More blood vessels means more vessel length, which means more resistance, which means we have to generate more blood pressure. Not a good sequence we got going on there. But this is not a quickly changeable thing like increasing or decreasing my number of and my length of blood vessels. So this one's not as good. But you know what is really good? Radius. Of course, remember from math class, you take a circle, you find the center. From the center to any point on the circle is the radius. And basically radius is a measure of, how big the tube is, right? Like how wide the tube is. And a narrower tube has more resistance. So as radius goes down, resistance goes up. There is an inversely proportional relationship there. Bigger radius, so bigger tube, lower resistance. Now here's the kicker. It's not a one-to-one relationship. Any change in radius, I got to take it to the fourth power to achieve that change in resistance. So if my radius decreases by 3, like a factor of 3, then my resistance increases by a factor of 3 to the fourth. And of course, math class, right? 3 to the fourth is 3. Times 3. Times 3. 3 times 3 is 9. Times 3 is 27. Times 3 is 81. So a threefold change in resistance yields an 81-fold change a threefold change in radius. There we go. One of these days I'm going to make a video with 0 mistakes. We'll see. A threefold change in radius begets an 81-fold change in resistance. So small change in radius can have a big change in resistance. Plus, radius is quickly changeable. There we go. Radius is quickly changeable. I can adjust the amount of muscle tone in my tunica media by releasing more or less norepinephrine, which we see here, or by adjusting the levels of local chemical controls, and I can get a quick change in resistance by changing radius, and therefore get a quick change in flow. All right, if we summed it all up, resistance is directly proportional to length and viscosity, and then inversely proportional to radius to the fourth power. So here's your question. Which of these tubes has the greatest? Which of these tubes has the least resistance? Figure that out for me. Let me know what you get. Now, if I want more blood to go to a place, I need to do one or two things. I need to change at least one of these, if not both. Ideally, if I want greater flow, I'm going to increase my cardiac output, which means more pressure, bigger pressure gradient, and then I'm going to vasodilate in the area where I want to send blood, lower the resistance there. So with a bigger gradient and lower resistance, I will get more blood flow. Similarly, if I want a decrease in blood flow, I'm going to lower my pressure gradient by lowering cardiac output and increase my resistance by doing some vasoconstriction. All right, this is great. Let's now pivot to controlling blood pressure. Short-term blood pressure control. Like the kind of control you need when you get up from a lying down position. When you get up from a lying down position, blood is going to kind of be pulled down by gravity. Pressure in your head starts to drop. We need to fix that. There are 3 brain centers that are super important for short-term blood pressure control. Those 3 brain centers are all located in my medulla oblongata. There is a cardioinhibitory center that we met already, a cardioacceleratory center that we met as well, and a vasomotor center. So cardioinhibitory centers, obviously, those are going to affect cardiac output. The cardioinhibitory and cardioacceleratory centers affect cardiac output. Cardioinhibitory center is going to decrease cardiac output and thereby decrease blood pressure. Activating the cardioacceleratory center is going to increase cardiac output and thereby increase blood pressure. The vasomotor center is going to affect your blood vessels. If you increase its activity, you increase the total peripheral resistance. And we know with more resistance, that's going to cause blood pressure to go up. Notice that vasomotor and cardioacceleratory are buddies. They are both having the same effect on blood pressure. In general, if I know what cardioacceleratory is doing, I know what vasomotor is doing, and it's the same thing, and vice versa. And whatever these guys are doing, cardioinhibitory is doing the opposite. Okay, let's expand on this a little bit. If I have more vasomotor center activity, I have more sympathetic signals to my arterioles. So that means I'm releasing more norepinephrine on that tunica media. Tunica media is contracting. Vasoconstriction, in other words, resistance is going up. Pressure is going up as well. All right, sweet. Let's look at the next one. If we turn on our cardioacceleratory center, more sympathetic activity signals to the heart, increased norepinephrine on the heart. So the SA node gets more norepinephrine, so heart rate goes up. The muscle in the heart gets more norepinephrine, so stroke volume goes up. When these two go up, we know cardiac output goes up because we remember that cardiac output is the product of heart rate and stroke volume. And if cardiac output goes up, blood pressure goes up too. Let's run through the third sequence. If I turn on my cardioinhibitory center, now I got more parasympathetic output to the heart. More acetylcholine being released onto my SA node. Heart rate goes down, cardiac output goes down, thus blood pressure also goes down. So those are the ways I change blood pressure for the short term. But I know what you're thinking. I know what you're thinking. Pretty amazing how I'm doing this video and I literally know what you're thinking right now as you're watching it. You are thinking, how do we measure the blood pressure? Am I right? Was that what you were thinking? You were like, how do we know when to turn these things on and off? All right. So there's this thing called the baroreceptor reflex. And baroreceptors are special cells. They're special cells and they are measuring something. Go back to chemistry or physics class and remind yourself what this prefix baro means. Baro means pressure. So there are special cells measuring pressure. Now they're measuring blood pressure. And I've got baroreceptors in my aortic arch. This makes total sense because blood in the aortic arch is basically going everywhere. And if blood is going everywhere, that's pretty darn important. I got to make sure it has enough pressure to do so. I also have baroreceptors in this little area called the carotid sinus. So remember from lab, we got a common carotid artery right here. This is the left common carotid. We know the word common means it's splitting. So this is my internal carotid here, and this is my external carotid right here. And at that splitting or slightly above it, there is a widening in the artery, and that wide area is called the carotid sinus. So there's this wide area of your carotid artery right at the split. And I've got baroreceptors there, which makes ridiculously perfect sense because where's the blood going? Blood is going to our brain. And we got to make sure that we have enough pressure in blood going to the brain. So this is how I'm measuring pressure. I'm using baroreceptors, which these are the two major locations for them. There are other locations as well, but these are the two major ones. And these locations make perfect sense. All right, good stuff. Let's talk about this reflex a little more. So here I've zoomed in on my aortic arch. So here's the aortic arch down here. The ascending aorta, descending aorta would be over here. I got special cells in that aortic wall that are my baroreceptors. Here is my brachiocephalic artery, my first branch off that aortic arch. This guy coming up here is my left subclavian. This guy right here is my left common, I meant right, I am sorry. Like I said, one of these days I'll make zero mistakes. Here's my right common carotid artery. Here's my left common carotid artery over here. And we know the common carotids are splitting to give us external and internals. And right at or above that split, we have a widening. And the widening is the carotid sinus. And I got baroreceptors there. So these baroreceptors are always measuring blood pressure. They're always sending signals up to the medulla. The aortic ones send signals via the vagus nerve, cranial nerve number 10. The carotid sinus receptors send signals via cranial nerve number 9, which you remember was GP, the glossopharyngeal nerve. And the frequency of these signals is proportional to the average pressure in these areas. So if blood pressure goes up, more signals go up to those cardiac and vasomotor centers, and then they adjust what they're doing in order to fix blood pressure. If blood pressure goes down, fewer signals travel, and these centers are going to adjust their output as well. So this is the baroreceptor reflex. Now you can actually test it. We'll talk about some, I'm not going to talk about some ways to test it. I'm going to keep on going. Try and get this video done without digressing too much. So another way we control blood pressure is this thing called the adrenal medullary mechanism, where our adrenal glands, which sit on top of our kidneys, release epinephrine and a small amount of norepinephrine in response to large drops in blood pressure, increases in physical activity, or stressful situations. Now, this epinephrine, if we release it when blood pressure drops, of course, it is going to make blood pressure increase. And that makes sense in physical activity. We want blood pressure to go up to drive blood flow to the muscles. And in stressful situations, we want blood pressure to go up for the same reason, to drive blood flow to our muscles, to our skin, to our lungs. So that's our adrenal medullary mechanism. So if you were faced with this grizzly here, you would definitely turn on that adrenal medulla. What would the epinephrine or norepinephrine do to all of these? So figure that out. I will give you a hint. There is going to be a theme here, okay, to your answers. So figure that out. Come talk to me about it. Yeah. All right. Another way we control blood pressure, and this isn't quite as short term, is this thing called the renin, angiotensin, aldosterone system. And I know you like it already. It's got like a triple name up there. It's pretty good. This is a kidney. The kidney is always releasing some renin, and renin is this chemical. Kidney is always releasing some renin. Renin comes from the kidney, so it's called renin. By the way, if you say renin, you're actually talking about something different that's made in the stomach with two N's. So don't say renin. Sorry, don't say renin, say renin. So your kidney is always making some renin, which is always causing you to produce some of another chemical called angiotensin 2. Now, what's the advantage of always making some? Well, if I'm always making some, I can then make more, or I can make less. I can go in either direction, and that's good for control. Now, always making some renin, there then results in the production of some angiotensin II. Look at this name. This is a beautiful name. Angiotensin. Angio means vessel. Tensin, we're thinking tension, like contraction. So angiotensin II, as its name suggests, no surprise here, is a vasoconstrictor. There's an angiotensin 1 as well. We'll talk about that later, but not today. Angiotensin 2 is a vasoconstrictor. So what does it do to resistance? It increases resistance. So what does it do to blood pressure? It increases blood pressure. So we're always making this chemical that is always keeping our blood pressure at a certain homeostatic level. And by always releasing some of it, not like going crazy in either direction, we can release more or we can release less. What a great situation. Okay, it's actually a little more complicated. Let's add some stuff. Because angiotensin II, I mentioned its vasoconstrictory nature that its name suggests, which increases total peripheral resistance, which increases blood pressure. But angiotensin also makes you thirstier. And if you're thirstier, I'm opening a bottle of water right now, and I just took a drink, and that drink of water is going to slightly increase my blood volume, which is going to increase my blood pressure. So angiotensin II is going to increase blood pressure not only by vasoconstricting, but also by increasing your thirstiness. And angiotensin II makes you release 2 hormones. Hey, angiotensin II makes you release 2 hormones. And they both start with the letter A. A lot of A's going on here. The 2 hormones are antidiuretic hormone and aldosterone. Let's just focus on one word here. Antidiuretic. That means against peeing. So this hormone stops you from peeing. And aldosterone, these guys are buddies, they're doing the same thing. Aldosterone also reduces your peeing. If I reduce my urine output, my blood volume is going to rise, especially when I couple that with thirstiness and increased drinking. And with more blood volume, I physically have more blood, therefore, I have more blood pressure. All right, that is my renin, angiotensin, aldosterone system. And now we got another chemical that impacts blood pressure. Of course, this is a heart. You know all the parts. Atria. When the atria stretch, they're going to release a chemical called atrial natriuretic peptide. Peptide is just like a short amino acid chain. It's like, think of it as like a tiny protein. So it's a tiny protein from the atria. I like to abbreviate atrial natriuretic peptide, ANP. So when the atria stretch, and maybe they're stretching, why might they be stretching? Let's think about that. Maybe they're stretching because there is an increase in blood volume, which goes along with an increase in blood pressure. So atria stretch, they release ANP, atrial natriuretic peptide. It is a vasodilator, which means it decreases peripheral resistance, which means it decreases blood pressure. ANP also increases urine output of sodium. If we're peeing more sodium away, osmotically we pee away more water. So there is just an increase in urine volume that's going on here. And if we pee away more water, less water remains in the blood, decreased blood volume, and that is going to result in decreased blood pressure. By the way, here's how I remember it. ANP, atrial natriuretic, ANP makes you, bear with me here, A and P makes you P and A. And by P, I mean P like urinate. So A and P makes you PNA. And if you pee away more sodium, you pee away more water, which means you lower your blood volume and lower your blood pressure. The natri part here means sodium. The uretic part here means urine. So natriuretic has to do with sodium and the urine. All right, beautiful. This is a blood pressure and blood volume lowering chemical here. We had some increasers previously. Nice to have a lowerer. And with that, I want to do a couple more things. Number one, I want to look at how blood velocity, the speed of blood flow changes as I go through my blood vessels. Notice how as we're close to the heart, the speed of blood is very fast. Then it decreases as we get to our capillaries, which is great for exchange, but then it actually increases as we go through our veins. And the reason for this shape here is because of the change in area. The slow flow in capillaries is because their area is so huge. So as a river gets wider, it slows down. As a river gets narrower, it speeds up. If you've never seen this, you need to go to a river and appreciate this. All right, go rafting or go kayaking, canoeing, all fun things. But lots of area means flow is slow. As area is smaller, flow is higher, faster. And of course, the presence of the pump at the beginning here has an impact as well. Notice that my area graph, the blue one here, looks more like an A. If I just draw a line right there, my area graph looks kind of like an A. And my velocity graph is like a very drawn out V. So if I spell velocity like that, look at that V. And I probably should have made it a little bit higher over here. There we go. Perfect. So we got to know that relationship. We pivoted to a random short topic there. And let's talk about one more thing. Blood distribution during exercise. When you are exercising, obviously you need increased flow where? To your muscles, your skeletal muscles, to power them, to give them the nutrients and oxygen they need, and to take away the wastes and the heat and the CO2. We also need more blood to go to our lungs, so we can do gas exchange. And we need more blood to go to our skin, so we can get rid of that heat we generate during exercise. So these are three important changes in blood flow that occur during exercise that we got to know. I bet you knew these already just intuitively. And with that, everyone, we are finally done this vascular system set of lectures. I am breaking things in my office here. And I will see you guys later on when we talk about the lymphatic system. As always, it's been great. See you later.